Okay, we can start our class. First, in our molecular cell biology chapter, the first unit is eukaryotic cytoskeleton. Uh, then first, we have to know what is eukaryotic cells. Uh, in our plus two graduation level, we know that what is the difference between prokaryotic and eukaryotic cells. My voice is audible or not?
Yes, sir. Yes,
Yes,
sir.
sir.
Yes,
Yes,
sir.
sir.
Yes,
Yes,
sir.
Audible,
sir.
sir.
Okay, okay. Thank you. In our plus one graduation level, as we know, what is prokaryotic and eukaryotic cells. Here we are describe about the eukaryotic cytoskeleton. Before going to the eukaryotic cytoskeleton, we have to know about the what is eukaryotic cells. If we go to the eukaryotic cells are complex cells and found in animals, plants, fungi, and protists. Unlike simpler prokaryotic cells like bacteria, eukaryotic cells have nucleus which act like a brain of the cells, which stores and protect the DNA, that is our genetic materials, and that controls everything the cells does. Every molecular level changes or any signaling occurs this through and this nucleus. These cells have membrane bound organelles. As we know, all the eukaryotic cells having different organelles, organelles are membrane bound organelles which are like tiny factories or workstations, each with specific job to keep the cell alive and functioning smoothly. If we go to the eukaryotic subcellular organelle, we found here the structure of eukaryotic cells. In eukaryotic cell, there are different subcellular organelles like nucleus, Golgi, endoplasmic reticulum, mitochondria, vacuoles. This is the plasma membrane and microfilament, lysosome, microtubule, cytoplasm. These are called subcellular organelles. What are these subcellular organelles function? If we go to the first subcellular organelle, that is nucleus. If we go to see the structure of nucleus, and these are membrane bound organelle and constituent the chromatin material, and this is the nucleoplasm. This is the cytoplasm of nucleus. This is called nucleoplasm, and this is a nuclear pore, and the cover membrane boundary is called as nuclear envelope. What is the function of this nucleus? It's like the control center of brain of the cells. It stores the genetic materials and coordinates activities like growth, metabolism, and reproduction. This is the main process which does the nucleus. And the key parts of the this nucleus is nuclear envelope. This is a double membrane with pores. From these pores, all the RNA are transferred to the cells, and different material from synthesized inside the nucleus are transported through these pores. And the nucleolus, this is the nucleolus. The major function of the nucleolus is it synthesize the ribosome, because ribosome required for our protein synthesis process. This is the function of nucleus. Then we go for mitochondria. uh, Sorry, mitochondria. As we know, mitochondria is called as powerhouse of the cells because it produces energy in the form of ATP during the, as we know, Krebs cycle or TCA cycle, that is tricarboxylic acid cycles, and it produces ATP. If you go to the structure of this mitochondria have uh, their own DNA, that is mitochondrial DNA, and the double membrane, that is outer membrane and inner membrane, and different types of uh, proteins and ribosomes are found inside the mitochondria. Then if you go to the sorry, I can- then the third subcellular organelle is endoplasmic reticulum. What is the function of endoplasmic reticulum? The endoplasmic reticulum is a manufacturing and packaging system of protein. After synthesis of protein inside synthesis of protein uh, in cytoplasm, then the protein is transported inside to the endoplasmic reticulum for folding. After folding, the protein is then manufactured and packaged inside the endoplasmic reticulum through different chapters. And this endoplasmic reticulum is classified into two types. One is rough endoplasmic reticulum and another one is smooth endoplasmic reticulum. This is the rough endoplasmic reticulum due to the presence of ribosome inside the rough endoplasmic reticulum, uh, above the rough endoplasmic reticulum. In the smooth endoplasmic reticulum, the ribosome is absent. This rough endoplasmic reticulum studded with ribosomes and makes and process the protein. It basically synthesizes the proteins and transported inside the ER. And smooth ER, endoplasmic reticulum, makes lipids. This is the important part. Smooth endoplasmic reticulum makes lipids and detoxify harmful substance that is called genolytic substance. Uh, um, this smooth endoplasmic reticulum having one protein that is called cytochrome P450, which detoxify all the genolytic substance inside the endoplasmic reticulum. And fourth one is Golgi apparatus. Uh, this Golgi apparatus or Golgi body, also called as post office box, because it sorted all the proteins to the different targeted organelles or outside the cells. And this Golgi apparatus, it can modify the proteins and sorts and packages all the proteins uh, and lipids made by the endoplasmic reticulum and then sends them to their destination. Suppose one is protein is targeted to lysosome. How it go to the lysosome? After packaging inside the Golgi body, it's sort or transported into the lysosome or mitochondria or peroxisome or outside the cell through these small vesicles. Then fifth one is lysosome. As we know, lysosome is called as cleanup crew of the cells. They contains different hydrolytic enzymes, and these hydrolytic enzymes damage different organelles or foreign invaders like bacteria. Like if a pathogen coming inside the cell, then this lysosome fused with that pathogen and damage that pathogen or digest them, and they form digest all the pathogen virus like particles. These are the major function of the this lysosome. Then sixth one is peroxisome. The major function of the peroxisome is this is breakdown of fatty acid and detoxify harmful substance like hydrogen peroxide. During the oxidative process, this hydrogen peroxide is produced, and this peroxidation having enzyme that cleaves this hydrogen peroxide to water and oxygen. This is the major function of the peroxisome. Then if you go to the ribosome, as we know, ribosome makes the proteins and some float freely in the cytoplasm. This ribosome both found in inside the rough endoplasmic reticulum, or some are found in the cytoplasm, and others. And if we go to the structure of like the ribosome, this ribosome having two subunits, one is larger subunit and another one is smaller subunit. In eukaryotic, this ribosome is 80S ribosome, one is 60S and another one is 40S. 40S means the S means Sedberg unit. During the centrifugal cycle, these two subunits are produced, and each subunit having different molecular weight according to them. This is 60S and another one is 40S. Then the another subcellular organelle, an important organelle, that is called cell membrane. This cell membrane is also called selective permeable membrane, and the function of this cell membrane is it's a protective barrier. that controls what enters and leaves the cell. It also helps in communication with other cell. Like if you go to the structure of the cell membrane, this is a phospholipid bilayer structure, and outer side the this is an extrinsic protein. These are intrinsic or integral proteins, and these are peripheral proteins. Like this, these during the signaling process, these extrinsic proteins are helps in communication with the other cell or diffusion of different gases or like signaling molecules through the this plasma membrane. Then another organelle is called centrosome or centrioles. These centrosome or centrioles help organizing microtubules and are important for cell division. We can study details in this microtubule process for this centrosome and centrioles during the cell division process. This is called centrosome, and inner side is called centrioles, and these are required during the cell cycle and cell division processes. Then another one is vacuoles. These vacuoles are store nutrient waste products and helps maintain turgor pressure in plant cells, but in animal cells have small vacuoles to reserve some waste materials, while plant cells have large central vacuoles. Basically, major of the vacuoles are found in plant cell, and in animals a small number are found because it stores only waste materials. But in plant cell, it stores nutrient waste products as well as maintain the turgor pressure. The another one, the last one is cytoskeleton. As we study today, the topic is eukaryotic cytoskeleton. This is the major process, major subcellular organelles, and this cytoskeleton act as cell's framework. It gives shape to the cell, offers support to the cell, and also movement of the cells. It's made up of microtubules, uh, intermediate filaments, and microfilaments. If you go to the structure of cytoskeleton, these are intermediate filaments, these are called microtubules, and these are microfilaments, which gives the shape, support, movement of the cells. These are the basic skeleton of our cell. Any doubt till here?
No, sir.
No, sir.
No, sir.
Sir, can you please share the PDF file?
Okay, okay. After my class, I will share the PPT.
Okay, sir. Thank you.
Then we can start with our microfilament, that is actin filaments. If you go to the microfilament structure, microfilaments also known as actin filaments.
Excuse me, sir.
Yeah,
Sir, what is the pattern for exam theory paper? The one question will be for 5 marks or 10 marks
you can contact the respective coordinator section.
because there is no previous year questions are not available in the site also.
Okay, I can talk with our coordinators. Then I'll.
Yes, sir. Okay, sir.
We'll discuss about the actin filaments or actin structure. This is called microfilaments, and the microfilaments also known as actin filaments. These are dynamic structure and composed of actin protein monomers, which polymerize into double-stranded helix-like structures and forming a 7 nanometer diameter filaments. These actin filaments is essential for the cell shape, movement, and intracellular transport uh, during the protein transport. If we go to the structure of these actin filaments, These of these microfilaments are built from these globular proteins. This is called as G actin, and these globular proteins are polymerized into filamentous protein. This is total called F actin, and this filament structure forms a double stranded. This is the double stranded helix like pattern with a diameter about 7 nanometer. And if we go to the polarity, what is polar? Polar means one end having plus charge and another one end having minus charge. If you go to the polarity of a microfilament, these microfilaments have a distinct polarity with a plus end. This is the plus end where the G globular actin or G actin is added, and another one end is minus end where the uh, minus and where the monomers are removed. This addition and remove of these microfilaments causes the contraction and expansion of these uh, microfilaments or actin filaments inside the cells. Then if we go to the major important function, that is network formation in cells, actin filaments are upon organized into network or bundles, and these interacting with other proteins to form a structure like stress fibers and the cell cortex. If we go to the dynamic of these microfilaments are highly dynamic and constantly undergoing polymerization. uh, that is called addition of this globular actin filament into in plus and depolymerization or shrinkage of this uh, the globular actin filament. This is called addition, and this is called polymerization, and this is removal of this G actin, and this is called uh, shrinkage of these microfilaments. Then, if we go to the actin monomer binding, G-actin monomers binds to the ATP molecules. It always G-actin monomer always require one molecules of ATP, which is necessary for polymerization of this G-actin. And the during the ATP hydrolysis, this ATP once incorporated into an actin filament, then this ATP is hydrolyzed, and which can influence the stability and dynamics of this filament, because the if ATP is not hydrolyzed, the polymerization and depolymerization will not occur. This is the major process of ATP hydrolysis. 1 minute, please. Sorry. Huh. If we go to the dynamic nature of these microfilaments, the microfilaments are highly dynamic and constantly undergoing polymerization or depolymerization. For polymerization and depolymerization, it requires one molecules of ATP, and this ATP molecule should be hydrolyzed. If this ATP molecule will not hydrolyze, then the polymerization or depolymerization will not occur, or the cell shape or size will not be maintained. And another function of these microfilaments is cell motility. Due to this dynamic nature of microfilaments, this allows cell to change shape, because this shrinkage and polymerization and depolymerization process, the cells is change its shape and move and perform function like muscle contraction, because during muscle contraction the polymerization and depolymerization occurs. And amoeboid movement, um, amoeboid movement means the movement of the cells because of this actin filaments. And the intracellular transport. Another function is intracellular transport. Microfilament can serve as a tracks for motor proteins like myosin. This facilitating transport of vesicles and other cellular cargo. Suppose we are studying the function of Golgi apparatus. Golgi is the post office box, and it sends the uh, vesicle-like structure from Golgi to nucleus, or Golgi to cytoplasm, or Golgi to mitochondria, or Golgi to peroxisome. How this occur? These vesicle is transported by the help of these microfilaments, by this polymerization and depolymerization. And by this polymerization and depolymerization process, the vesicle is transported from Golgi to the respective different subcellular organelles. If we go to the cell shape and support, microfilaments are crucial for maintaining cell shape and providing structural support, especially in the cell cortex. Because we'll describe this cell shape and support. Uh, and another function is cross-linking protein. Microfilaments with different proteins like fibrin, actinin, spectrin, dystrophin, and filamin help organize actin filament into various structure and network. And this network helps the skeleton of the cells or inside the cells. This protein is essential. If we go to the organization of this cortical cytoskeleton, this is the organization of the this cortical cytoskeleton to hold the how it holds the plasma membrane. If we go to the cortical cytoskeleton or actin cortex, this is called actin cortex, and is a specialized layer of proteins located beneath the plasma membrane, and this crucial for cell shape. This is required for cells shape because it is found beneath the plasma membrane to hold the plasma membrane to make a particular shape of these eukaryotic cells. Um, and stability and various cellular processes like cell division and migration. For migration, this actin filament also required. And location and composition of the this cortical cytoskeleton is a specialized region of the cytoskeleton found just beneath the plasma membrane and forming a network like proteins, different star network like proteins, and these proteins are bound to this plasma membrane to retain its shape. And the key function of this cortical cytoskeleton is structural support and stability. It provides a structural support and mechanical stability because during this metabolical process, this shape of the cell will not change, and this gives the mechanical stability to the cell membrane, or contributing the cell shape and integrity of our eukaryotic cell. Then cell migration and motility, it helps in measure, it helps in uh, a cell movement and migration by facilitating change in cell shape. Because if we change the cell shape, this cortical cytoskeleton is required to change the cell shape. And if we go to the adhesion, this cortical cytoskeleton is required for communication with another cell for the adhesion. Then cell division. During the cell division from one cell to another, this cortical cytoskeleton is required for cell division and forming a contractile ring structure. We'll discuss about this contractile ring structure during cell division process in third unit, and that separates the two daughter cell. It separates daughter cell, and it also helps the organelle transport. It helps the transport of organelles within the cell during the metabolic or cytoplasmic disturbance. It will also transport the different organelles to one part to another inside these eukaryotic cells. And the modulation of membrane behavior, it also modulate the membrane behavior, because we know plasma membrane is a fluid mosaic model, and it acts as the modulator of the membrane behavior and cell surface property, and it also changes the cell surface. These are the major function of this cortical cytoskeleton. If we go to the biological significance of this actin cytoskeleton and myosin, and this actin cytoskeleton and myosin are crucial for cell structure, movement, and various cellular processes, acting as a dynamic network that generates forces for cell motility, division, and intracellular transport. These are required for the cell motility. The cytoskeleton is required for cell motility and during the cell division process and intracellular transport like vesicular transport or protein transport during the metabolical process. If you go to the actin cytoskeleton and their biological significance, the actin cytoskeleton is a network of actin filaments. Uh, we are described about this actin filament. This is called microfilament that provides structural support. Basically, this cytoskeleton provide the structural support, shape, and mechanical strength to the cells. If you go to the function of this actin cytoskeleton, it provides different cell shape, stability. Uh, this filament network form cell shape and stability and cell motility during the cell movement. It also help from uh, for this cell movement process, including crawling, migration, and engulfing particle. During engulfing process, for an example, one bacteria is outside the cells, then it engulfs that bacteria and it will fuse to the lysosome by this by the help of this cytoskeleton. This cytoskeleton provide a group, like if you go to the structure, you will find like this group. This is called endocytic process. In this process, this cytoskeleton also help to form these endocytic vesicles, and these endocytic vesicles will fuse with this lysosome to kill that bacteria. Then during the cell division process, this also forms a contractile ring-like structure to separate during the anaphase process, and the intracellular transport. This actin filament also, along with the myosin motors, facilitate the movement of organelles and vesicle within the cell, and also help in signal transduction. This actin cytoskeleton can transduce signal from external environment to the inside of the cells, influencing cell behavior. For an example, during any signal transduction process, these cytoskeleton molecules required uh, for the recognition of different like cells, different stimuli. Then we go for the myosin structure. If we go to the structure of myosin, myosins are family of motor proteins. This is called structure of myosins. And if we go to the myosin molecules, and these myosins are family of motor proteins, and that converts chemical energy, that is ATP, into mechanical energy, generating force and movement. This is the major motor protein during the muscle contraction. This is called myosin. And the if you go to the function of this myosin are best known for their role in muscle contraction, where they interact with actin filament to generate force and movement. These myosin heads are bind to these actin filaments and utilize one ATP molecules. By this polymerization and depolymerization reaction, after addition, um, uh, in placent, the G protein G actin is added. This is called polymerization, and this is called depolymerization. Here also called polymerization and depolymerization. This myosin head bind to this actin filament and utilizes ATP molecules and form a sliding filament like structure. This is called sliding filament theory for this muscle contraction. And these myosins are best known for their muscle contraction process, where they interact with these actin filaments, and helps in cell motility. Also, myosins are involved in various types of cell motility, including cell migration, crawling, and the movement of the organelles and vesicles. Because of this, with these actin filaments and actin and myosin, these are the major protein are required for the this muscle contraction process, or transport of different vesicles, and migration of different cells. Then myosin also play a role in cell division and assisting in the formation of the contractile ring and separation of the chromosome. They also separate the chromosome by their mechanical force. And intracellular, it also myosin are involved in the transport of organelle and vesicle within the cell, along with the actin filaments. If we go to the main structure of this myosin, these are two dimeric structure and having a coiled domain, and these are the tail is non-helical coil-like structure, and this is required during this muscle contraction process, and this is called sliding filament theory. Any doubt till this process?
No, sir.
No, sir.
No, sir.
Okay. Then we go for the another cytoskeleton. Uh, okay.
Hello, sir.
The,
Sir, एक
yes.
क्वेश्चन है. Sir, if
बोलिए.
ATP is not hydrolyzed, then क्या polymerization और depolymerization नहीं होगा?
नहीं 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 होगा. Energy is
Sir,
required during this
reason?
during your running process, or if you are walking, energy is utilized. If you have not having any uh, ATP molecules or energy, you cannot walk or run. Because of this polymerization and depolymerization, this sliding is occur. One is polymerization and depolymerization. If you add here and minus here, if you add here and minus here, this will go forward and this will go forward. This is called sliding of this. This will go this way, this will go this way. This is called contraction, and during this, this is relaxation. By this process, the muscle contraction will occur. And because these ATP molecules help in forming a dimer and filamentous like a structure during the formation of the F-actin molecules. Minimum energy is required for dimerization and filamentous like structure formation.
Thank you, sir.
Then, if you go to the actin and myosin interaction, myosin work along with actin filament and using ATP hydrolysis to generate force and movement, and this is required for hydrolysis of one molecules of ATP. The interaction between the actin and myosin filament is essential for generating the forces required for cell motility, division, and other cellular processes like protein transport or cell division. And the interaction between the actin and myosin is tightly regulated, allowing cells to control their movement and change their shapes. If we go to the biological significance of these two, that is actin and myosin. The actin cytoskeleton and myosin are essential for maintaining cell shape and structure, and these proteins are crucial for cellular movement, allowing cells to migrate, crawl, or engulf different like particles to form endocytic vesicles. And these actin and myosin are also essential for cell division, ensuring proper separation of chromosome and the formation of two daughter cells. And these two proteins facilitate the movement of organelles and vesicle within the cell, and this enable efficient cellular function. And the major important point is the these two proteins having different pathological significance, like dysregulation of this actin cytoskeleton and myosin can contribute to various diseases including cancer, neurodegeneration, and cardiovascular diseases. These two protein are most important protein for requirement of for shell movement or cell migrate or crawl or engulf different particles or any types of cellular function or involvement of these two actin and myosin interaction is required. Then another cytoskeleton is microtubules. If we go to the one is
Excuse
actin,
me, sir.
yeah,
Excuse me, sir. Can you
yeah,
please tell us something about rigor mortis?
rigor.
Rigor mortis.
Okay. Uh, this polymerization help in this actin and myosin or actin polymerization help in a rigor mortis, that is a rigid structure. This form a rigid structure. If you during rigor mortis during the in which process like if one part if was one cell having one part of the cell is totally dead, uh, we declare that part is dead because how will call that part is dead because of this rigor mortis, that is rigidity. During this dead part, the actin and myosin filaments are totally dead or dysfunction of this actin and myosin proteins. And if this protein structure is destabilized at that part, and we call that part is called rigor mortis. Understood?
Yes, sir.
A rigidity, rigid structure.
If a small part of our body suffered from the rigor mortis, can we recover from that, or what will happen after that?
If good question. Uh, if we found one part is dead because of this our cell having different chaperone proteins to help during this stress condition, like any stress condition like heat stress, cold stress, any stress condition or shock. These proteins are activated to help these actin and myosin proteins to form a to balance its structure, or these are called heat shock proteins. This HSP family stabilize the different important protein structure. If these protein structure are not replaced or properly repaired, or their entropy or energy is not properly maintained, and the fate of that protein will be degraded. Ubiquitin tag will be attached to that protein, and that protein part is degraded, and this part is called rigor mortis. This cannot be replaced. Okay.
Okay, sir. Thank you, sir.
Then we go for next cytoskeleton, that is called uh, microtubule. If we go to the microtubule, microtubules are dynamic hollow cylinder like structure and composed of major two protein. One is alpha and beta tubulin, and these alpha and beta tubulins are heterodimers. Uh, in actin, that these two proteins are G proteins are homodimers, but here alpha and beta tubulins are heterodimer, and these are essential for mm, cell structure, organization, and movement, including chromosome segregation during mitosis and meiosis process. These microtubules help in chromosome segregation and intracellular transport. For an example, as we know, the ploidy of the chromosome, like Down syndrome, like extra number of chromosome or minus number of chromosome, 46, that is 47 or 45. By this, if the microtubule structure is damaged, and this will cause 46 or 47 like different chromosome. One chromosome is go to the extra number. By this help of this, if damage of this microtubular structure, and this microtubular structure is very hollow cylindrical like structure, and this having alpha and beta tubulin, and this help in cell structure, organization, and movement, including chromosome segregation and intracellular transport. If we go to the structure, these tubulin dimers, these microtubules are built from this alpha and beta tubulin, and this because this is heterodimers, not homo and hetero. What is the difference between homo and hetero? Homo means same molecular weight, hetero means different molecular weight. These two alpha and beta tubulin having different molecular weight, and this dimer polymerized into uh, protofilaments, which then uh, associate laterally to create a this like hollow like structure or cylindrical microtubules. But in actin, that is filamentous structure, here here polymerized into protofilament like structures, and these protofilaments will form a hollow cylindrical like microtubular structure. If we go to the polarity, actin filament also having polarity, having plus end and minus end. Here also in microtubular structure, you can find the one is plus one and another one is minus one. This plus and minus is required for polymerization and depolymerization of these microtubules. And if we go to the diameter, in actin filament, that is 7 nanometer in diameter, but here in microtubular structure, microtubules are approximately 25 nanometer in diameter, and inside the hollow cylinder, that is 14 nanometer, and having 13 protofilaments. In the 13 protofilaments of alpha and beta tubulins. This is the basic structure of these microtubules. Then if we go to the organization, these microtubules are major component of the cytoskeleton along with microfilaments. Always the microtubule and have microfilaments. These are the major components, and also intermediary filaments. And these microtubules form a microtubular organizing center inside the such as centrosome, where minor ends are anchored. And if you go to the dynamic of the microtubules exhibit dynamic instability, constantly undergoing cycles of rapid growth, polymerization, and shrinkage. In microtubule, uh, polymerization and depolymerization simultaneously occurs. If we go to the microtubule associated proteins, maps are regulated through microtubule dynamics, like polymerization and depolymerization, and organization and interaction with other different cellular structures. And this microtubule organization varies depending on cell type and functions, influencing cell shape, polarity, and intracellular transport. With the help of microfilaments and this transport different proteins or different vesicles inside the cells or cell to outside the cell. Um, if we go to the dynamics, how this polymerization and depolymerization occurs, and for this polymerization and depolymerization, microtubule dynamics require hydrolysis of GTP molecules. In actin filaments, that require ATP molecules. Here we require GTP molecules, that is guanosine triphosphate by beta tubulin. Here beta tubulin always required one molecule tubulin dimer bound to the GTP and tubulin dimer bound to the GTP. Always one molecule of GTP hydrolysis occurs and leading to changes in tubulin conformation and microtubule behavior. After the binding of this GTP molecule, this conformation and behavior will change. And this microtubule can rapidly grow by adding tubulin dimers to fluorescent and shrink by losing dimers, a process known as dynamic instability. Always addition and subtraction of this microtubules dimers will occur in plus and minus end to form a cylindrical like structure. And in the help of different motor proteins like kinesin and dyneins, these microtubules are tracks for intracellular transport and organelle movement. For the movement of inside the organelle with the microtubule help of the different proteins, that is kinesin and another one is dynein, for movement of this organelle inside the cells. During the cell division, microtubules form mitotic spindle because this mitotic spindle is required for proper separation of chromosome and ensures accurate segregation. This mitotic spindle is mm, most important part during this cell division process for correct separation of chromosome inside the cells during cell division. Then, if you go to the microtubules in cell shape and mitosis process, then microtubules are the key components of the cytoskeleton. This play a crucial role with the help of microfilaments also in both maintaining cell shape and facilitating accurate chromosome segregation during mitosis process, and forming the mitotic spindle to and guiding chromosome movement. For the movement of chromosome for centromere to the central part or central part to the segregation of this chromosome, 46 number of chromosome to 23 and 23 by this mitotic spindle. And if we go to the cytoskeletal framework, microtubules along with other cytoskeletal element like actin, this is microfilaments and intermediate filaments provide major is cytoskeletal framework structure framework, and that determines cell shape and organization and different organelle positioning such as inside intracellular transport of different cellular organelles, uh, and other cellular components are positioned correctly within the cells. If you go to the structure of this cell, these microtubules are helping proper organization of this nucleus, endoplasmic reticulum, Golgi body, vacuoles, mitochondria, peroxisome, all are properly organization by the help of this cytoskeletal protein that is microtubules and microfilaments. Then microtubules are, as we described, the microtubules are very dynamic because rapidly grow and shrink because of this polarity, that is plus and minus n. And during the mitosis, the microtubule are reorganized to form mitotic spindle. This is the very important point we describe during uh, the process of cell division, because there is a called spindle assemble checkpoint. We'll discuss it during cell cycle. This spindle assemble checkpoint, uh, by the help of this mitotic spindle, a structure responsible for separating of chromosome. Properly, chromosome is separated or not into two different daughter nuclei. And this chromosome segregation, the mitotic spindle uses microtubules to attach to the chromosome, that specialized region called kinetochores, and then uses motor proteins, that this is kinesin and dynein, to pull the chromosome apart, ensuring each daughter cells receive a complete set of chromosome or not. And the dynamic instability of this microtubule, where they rapidly grow and shrink, is crucial for the accurate formation and function of this mitotic spindle. And during the kinesis process, this microtubule also play a role in cytokinesis, the final stage of cell division, where the cell divides into two different daughter cells, and that help to position and guide the contractile ring to separate the two different daughter cells. This is the major part of the microfilaments, that is actin filaments, and this is uh, uh, microtubules. And these actin and microtubules, the major function is to maintaining the shape and size and mechanical strength to the eukaryotic cytoskeleton, eukaryotic cell cytoskeleton. Any doubt till here?
No doubt, sir.
Okay. ବୁଝି ହଉଛି ନା ବୁଝି ହଉନି?
ଆଜ୍ଞା ବୁଝି ହଉଛି sir.
Okay.
ସାର୍ ଆମର ଯୋଉ ସ୍ପିଣ୍ଡିଲ ଅଛି ସେଟା ସେଲ୍ ସାଇକେଲ ସ୍ପିଣ୍ଡିଲ ଫାଇବର୍ସ କୁହନ୍ତି ନା?
Spindle fiber. We'll discuss about the spindle assemble checkpoint, how the microtubule help in this sac formation, spindle assemble checkpoint for segregation of proper segregation of chromosome during the cell division, or say, or the formation of two different daughter nuclei.
Sir ଆମର further doubts ରହିଲେ sir ଆମେ ମାନେ କେମିତି କ୍ଲିୟର କରିବା ପାଇଁ ଷ୍ଟଡି ସେଣ୍ଟର ଯିବାକୁ ପଡିବ ନା ଆପଣଙ୍କୁ ଫୋନ କରି ବୁଝିବାକୁ ପଡିବ କେମିତି?
ମତେ ପଚାରି ପାରିବ।
ଆଜ୍ଞା ଆପଣ sir ଗ୍ରୁପରେ ଅଛନ୍ତି sir?
ନା ନା ମୋ ଗ୍ରୁପରେ ନାହିଁ।
ଆପଣଙ୍କ ନମ୍ବରଟା sir?
ହଁ ମୋ କ୍ଲାସ୍ ସରିଲେ
ଆଜ୍ଞା ଆଜ୍ଞା ଆଜ୍ଞା।
Okay, then we go for the last one, that is intermediate filaments. uh, we described the microfilaments and microtubules, and last one is intermediate filaments. If we go to the structure of this intermediate filaments, these are type of cytoskeletal protein that provides mechanical strength and stability to cells, and forming a network that resists tension and stress. This is very important, like a rope This intermediate filament formation like a rope. This gives a tension and stress. During the tension or stress, these intermediate filaments will help to maintain the cell, and they are composed of uh, various protein including keratin. If you go to the structure of skin, the skin is made up of keratin proteins, and these keratin proteins form a intermediate filaments, and these intermediate filaments to retain or resist different tension of our skin and different stress during every time skin is undergo different type of stress, and these intermediate filaments will help them maintain the stress and different types of tensions, um, to maintain the our skin cells healthy. And they are composed of various proteins, as we discussed by this is keratin, vimentin, and laminin, and organized into this rope like structure. This intermediate filaments having one N terminus and C terminus having a monomer like structure, and this is 45 in nanometer, and it will form a dimer structure that is 45 nanometer. And these two dimer to form a tetramer, and that is 60 nanometer structure, and these tetramers will wrap around each other a unit of length filament, that is ULF, that is 60 nanometer. And this 60 nanometer different 60 nanometer of unit length filament form a filamental like structure that is greater than 240 nanometer, uh, 240 nanometer like structure, which is called intermediate filaments. And these filaments are most important for this tension and stress of our cell. And if you go to the composition, these intermediate filaments are made up of diverse group of proteins, including keratin. Keratin is for epithelial cells, and vimentin is for mesenchymal cell, and laminin in the nuclear envelope. Uh, and this intermediate filaments protein monomer has a coil structure. This is called coil structure. And when the two monomers bind to form a dimer, this is the dimeric structure. One monomer and another monomer both form like a rope like structure, and this rope like structure gives a tensile strength to the uh, intermediate filaments. And these two dimers then associate end to end form a uh, this one end and another one end to form a tetramer, which is a basic building block of this intermediate filament. This is the basic building block. And the rope-like structure, this rope-like multiple tetramers assemble laterally to form a rope-like filament, which is the characteristic structure of this intermediate filaments. And these intermediate filaments have a diameter around 10 nanometer, which is larger than microfilaments but smaller than micro tubules. So this is called intermediate filament between the microfilament and microtubules. So the naming is intermediate filaments. Then if we go to the organization, intermediate filaments are part of the cytoskeleton, a network of the protein filaments that extends throughout the cytoplasm and provides the structural support and organization to the cells, because this rope-like structure gives a structural support and organization to the cells. And laminins, a type of this intermediate filaments, form a meshwork beneath the inner nuclear membrane, providing structural support and organizing chromatin. Because laminin is very important for organization of this uh, DNA molecules or different chromosome structures by the help of this intermediate filaments. And intermediate filaments also help maintain cell shape and stability, especially in tissues that are subjected to mechanical stress. We described how this rope-like structure gives the tension uh, or stress. During the stress, how it will help give mechanical strength. Mechanical stress such as skin and muscle. Maximum time our skin and muscles are in mechanical stress. So this intermediate filament help to maintain the structure of this skin and muscle by the help of these intermediate filaments. If we go to the different function of these intermediate filaments, these intermediate filaments provide mechanical strength and stability to the cells. This intermediate filament required for this mechanical strength or tensile strength and stability to the cells, and helping them withstand with mechanical stress and tension, because maximum time our muscle and the skin is undergone mechanical stress or tension. This mechanical stress and tension is properly maintained by these intermediate filaments. Then, if we go to the shape and integrity, this play a role in maintaining the cell shape and integrity, especially in tissues that are subjected to mechanical strength. And these intermediate filaments also anchor cells to each other and to the extracellular matrix, contributing to the tissue organization. Uh, this cell will form the tissue by the help of these intermediate filaments. And this laminin, a type of intermediate filaments, provide structural support to the nuclear involved and that organize chromatin. If we go to the structure of this involve, this is guarded by the laminin proteins, and these laminin proteins maintain the nuclear
ପୁରା
involved structure and the chromosome or DNA molecules. Then if we go to the intracellular signaling, this intermediate
ପୁରା
filaments
ହିଁ
can also
କାଲି କାଲି
participate.
କାଲି
Please mute. If you go to the intermediate filaments, this participate in intracellular signaling pathways and the influencing cell behavior and function. This also participate in intracellular signaling, like uh, different stimuli, like our skin molecules sense different stimuli like heat stress, cold stress, or any injury type of stress. This also gives an intracellular signaling uh, by this intermediate filament. These are the major component of our eukaryotic cytoskeleton, that is microfilaments, intermediate filaments, and the microtubules. Any doubt?
No, sir.
No, sir.
Good evening, sir.
Good evening.
Sir, ଯେ କ୍ୱେଶ୍ଚିନ୍ ଆସିବ ଏକ୍ଜାମ୍ର ପ୍ୟାଟର୍ନଟା ଟିକେ ଖାଲି ଜଣେଇ ଦେଇଥିଲେ ଭଲ ହେଇଥାନ୍ତା। ମାନେ ଆପଣ ଯେ ଏଇ ଚାପ୍ଟରରେ ଯେମିତି ବୁଝେଇଲେ ଲଙ୍ଗ କ୍ୱେଶ୍ଚିନ୍ ଟାଇପ୍ରେ ଆସିବ ନା ସର୍ଟ ନୋଟ୍ସ ଭାବରେ, ନା କେମିତି କ'ଣ ପ୍ୟାଟର୍ନ ଟିକେ ଜଣେଇ ଦେଇଥିଲେ ଭଲ ହେଇଥାନ୍ତା।
କାଲି ମୁଁ ପଚାରିକି କୋଅର୍ଡିନେଟର ସାର୍ଙ୍କୁ ମୁଁ କହିଦେବି ପ୍ୟାଟର୍ନ କ'ଣ ଆପଣଙ୍କର କ୍ୱେଶ୍ଚିନ୍ର ପ୍ୟାଟର୍ନ କ'ଣ।
ଥ୍ୟାଙ୍କ ୟୁ ସାର୍, ଥ୍ୟାଙ୍କ ୟୁ।
गुड इवनिंग सर।
ଗୁଡ୍ ଇଭିନିଂ।
ସାର୍ ଆପଣ ନୋଟ୍ସ
ସାର୍ ଆମର
ସେୟାର
ଆସାଇନମେଣ୍ଟ
କରିଦେଇଥିଲେ, ଟିକେ ଆମେ ପଢିକି କିଛି ଡାଉଟ୍ସ ଥିଲେ କାଲି ପଚାରି ଦେଇଥାନ୍ତୁ।
ହଁ ମୁଁ ଏଇଟା ପଠେଇଦେବି, ସରିଲେ ପଠେଇଦେବି
ଆଜ୍ଞା ସାର୍,
ଆପଣ।
ଠିକ ଅଛି।
ସାର୍ ଆମର ଆସାଇନମେଣ୍ଟ ର ଲାଷ୍ଟ ଡେଟ୍
ଆଜ୍ଞା
ଟା
ସାର୍।
ଟିକେ ଜାଣିଛନ୍ତି ସାର୍?
ନା ନା ମତେ ଇନଫର୍ମ କରାହେଇନି। ଆପଣ ଟିକେ ରେସ୍ପେକ୍ଟିଭ ସମସ୍ତଙ୍କୁ ଯୋଉ ସାର୍ କୋଅର୍ଡିନେଟର ସାରଙ୍କ ନମ୍ବର ନାହିଁ କି ଆପଣଙ୍କ ପାଖରେ?
ନାହିଁ ସାର୍ ନମ୍ବର ନା ତ।
କୋଅର୍ଡିନେଟର ସାରଙ୍କୁ ଟିକେ ପଚାରି ଦିଅନ୍ତୁ, ନହେଲେ ମୁଁ ସାରଙ୍କୁ କହିବି। ଆପଣଙ୍କର ଗ୍ରୁପ ଅଛି କହୁଛନ୍ତି ବା?
ସାର୍ ମାର୍ଚ୍ଚ 31 ଭିତରେ ପଠେଇବାକୁ କୁହାଯାଇଥିଲା। 15 ରୁ 31 ଭିତରେ ଆସାଇନମେଣ୍ଟ ସବମିଶନ ଥିଲା।
ହଁ ତ ଡେଟ୍ ତ ଜଣା ଅଛି, ତା ଭିତରେ ଆପଣ ଆସାଇନମେଣ୍ଟ ସବମିଟ କରିବେ।
କୋଅର୍ଡିନେଟର ସାର୍ ନମ୍ବର ଟା ସାର୍ ସେୟାର କରିଥାନ୍ତେ ଟିକେ।
ମୁଁ ପଚାରିକି ସାରଙ୍କୁ ସେୟାର କରିବି।
ଆଜ୍ଞା ସାର୍, ଥ୍ୟାଙ୍କ ୟୁ ସାର୍।
ମୁଁ ମୋ ମୋବାଇଲ ନମ୍ବର ସେୟାର କରିଦେଇଛି ମେସେଜରେ।
ଆଜ୍ଞା ସାର୍।
ଦେଖିଦିଅନ୍ତୁ। ମତେ ସେଇଥିରେ ପିଙ୍କ୍ କଲେ କିଏ ମୁଁ ପଠେଇଦେବି, ଆପଣଙ୍କ ଗ୍ରୁପରେ ଆପଣ ସେୟାର କରିଦେବେ।
ଆଜ୍ଞା ସାର୍, ଥ୍ୟାଙ୍କ ୟୁ ସାର୍।
ଆଉ କାହାର ଡାଉଟ୍ ଅଛି କଣ? ସିଲାବସ ର
ସାର୍ କାଲି ଯୋଉ କ୍ଲାସ ଅଛି ଆପଣ ହିଁ ନେବେ ନା ଅନ୍ୟ କିଏ ନେବେ?
ନା ନା ମୁଁ ନେବି, ଏବେ କଣ୍ଟିନ୍ୟୁଅସ 4ଟା ନେବି।
4ଟା ଦିନ?
ହୁଁ
ହଁ ସାର୍ ଠିକ ଅଛି
କାଲି
ସାର୍।
ସେଲ୍ ଟ୍ରାନ୍ସପୋର୍ଟ ଅଛି। ସିଲାବସ
ସାର୍ କିଛି
ଦେଖିଛନ୍ତି ନା
ଇମ୍ପୋର୍ଟାଣ୍ଟ
ସିଲାବସ
କ୍ୱେଶ୍ଚିନ୍
ଦେଖିନାହାନ୍ତି?
ସେୟାର କରିଥାନ୍ତେ।
ସିଲାବସ ଦେଖିଛନ୍ତି?
ଦେଖିଛୁ ସାର୍ ଆଜ୍ଞା।
ହୁଁ
ନାଇଁ ସାର୍ ସିଲାବସ୍।
ସିଲାବସ ଦେଖନ୍ତୁ, ସେୟାର କରିଛି ଦେଖନ୍ତୁ।
ସାର୍ ଟିକେ ସିଲାବସ ଟା ଗ୍ରୁପ ରେ ସେୟାର କରିଦେଲେ ଭଲ ହୁଅନ୍ତା।
ମୁଁ ଗ୍ରୁପ ରେ ନାହିଁ ନା, ମୁଁ କାହାକୁ ସେୟାର କରିଦେବି, ଆପଣ ଆପଣଙ୍କ ଭିତରେ ସେୟାର କରିଦେବେ।
ଆଜ୍ଞା ଆଜ୍ଞା, ଥ୍ୟାଙ୍କ ୟୁ।
ସାରଙ୍କୁ ମେସେଜ କରିଦେଲେ ସିଏ ପଠେଇ ଆପଣ
ହୁଁ
ଗ୍ରୁପରେ
ହୁଁ
ପଠେଇ।
ହୁଁ। ଏଇଟାରେ ଯୋଉ ଅଛି ୟୁକାରିଟିକ ର ସେଲ୍ ର ଷ୍ଟ୍ରକ୍ଚର ଆଣ୍ଡ ଫଙ୍କସନ, we describe about this structure and function. Then actin filament structure dynamics we also described. Organization of cortical cytoskeleton also described. Then actin cytoskeleton in cell shape and intercellular motility, cell locomotion and biological significance of this actin filament and myosin, microtubules and intermediate filaments. This is the molecular cell biology syllabus block 1. Then tomorrow we'll discuss about this cell transport.
ସାର୍ ଆମେ ଯଦି ଏଇଥିରୁ ଲଙ୍ଗ କ୍ୱେଶ୍ଚନ ପ୍ରିପେୟାର ହବୁ ତ
ହୁଁ
କୋଉଠୁ ପଢିବୁ ସାର୍? ବୁକ୍ ରୁ ପଢିବୁ ନା ଆପଣ ଯୋଉ ପିଡିଏଫ୍ ସେୟାର କରିଛନ୍ତି ସେଥିରୁ ପ୍ରିପେୟାର ହବୁ?
ମୁଁ ବୁଝେଇଦେଲି, ତମେକୁ ଯୋଉ ଦିଆଯାଇଛି ତମକୁ ନୋଟ୍ସ ଦିଆଯାଇଥିବ ନା?
ସାର୍
ନା
ଆମର
ଦିଆ
ବୁକ୍ ଅଛି।
ବୁକ୍ ଅଛି ନା?
ଆଜ୍ଞା।
ହଁ ସେଇ ବୁକ୍ ରୁ ଖାଲି ଜଷ୍ଟ ତମେ ଯୋଉଟା ବୁଝିଲ ନା ତାକୁ ଥରେ ରିଡ୍ କରିଦିଅ, ତମେ ବୁଝିପାରିବ। ଆଉ ଯଦି ତମେ ରିଡ୍ ନ କରିବ ତମେ ବୁଝିପାରିବ।
ଓକେ ସାର୍।
ତ କିଛି
ଏତିକି
ସାର୍
ଇମ୍ପୋର୍ଟାଣ୍ଟ
ସାର୍ ଏ
କ୍ୱେଶ୍ଚିନ୍
ସିଲାବସ୍
ଯେମିତି
କୋଉଠୁ
ଏକ୍ସାମ ପାଇଁ।
ଡାଉନଲୋଡ କରିପାରିବି?
ଶୁଣନ୍ତୁ, ଆ ଦେହରେ ତ ଟୋଟାଲି 3ଟା ହିଁ କ୍ୱେଶ୍ଚିନ୍। ଗୋଟେ ହେଇଛି ୟୁକାରିଟିକ ସେଲ୍ ଷ୍ଟ୍ରକ୍ଚର ଫଙ୍କସନ, ଗୋଟେ ଆକ୍ଟିନ ଫିଲାମେଣ୍ଟ, ଆଉ ଗୋଟେ ମାଇକ୍ରୋଟ୍ୟୁବୁଲ, ଆଉ ଗୋଟେ ଇଣ୍ଟରମିଡିଏଟ ଫିଲାମେଣ୍ଟ। ଏଇଆକୁ ତ ସଫଲ କରିକି ପକେଇବେ, ଆଉ କଣ ଆଥିରୁ କ୍ୱେଶ୍ଚିନ୍?
ସାର୍ ଆକ୍ଟିନ୍ ଫିଲାମେଣ୍ଟ ଥିୟରି
ଖାଲି
ବି ଇମ୍ପୋର୍ଟାଣ୍ଟ ଅଛି।
ପ୍ୟାଟର୍ନ ଟା ମୁଁ ଖାଲି ଜଣେଇଦେବି ଆପଣଙ୍କୁ କୋଉ ଟାଇପ ର ପଡିବ।
ଆଉ ସାର୍ କ୍ଲାସ ଟା 5:30ରେ ଷ୍ଟାର୍ଟ ହେଲା ନା? ଆଜି ମୁଁ 5:30ରେ ଜଏନ୍ କଲି, ସେ ମେଲ ଆସିଥିଲା 5:30 ତ, ମାନେ କନଫର୍ମ ହେବା ପାଇଁ ଟିକେ ପଚାରୁଥିଲି।
ହଁ ହଁ ସବୁଦିନ
5:30
5:30 ଏବେ, ଆଉ 3 ଦିନ 5:30।
ହଁ ହଁ ଠିକ ଅଛି ସାର୍।
ସାର୍ ଏକ୍ଜାମ ରେ ବୁକ୍ ଭିତରୁ କ୍ୱେଶ୍ଚନ ଆସେ ନା ବାହାରୁ ବି ଆସେ?
ନାଇଁ ନାଇଁ, ଏଇ ଯୋଉ ସିଲାବସ ଟା ମୁଁ ଦେଖେଇଲି ନାଇଁ, ଆରି ଭିତରୁ ହିଁ ଆସିବ।
ମାନେ आउटसाइड ରୁ କିଛି क्वेश्चन आसेनी सर?
ना ना आउटसाइड रू किसी आसबनी एवट्रेस जेतिके पढ़ा हला ना एतिके देखो ମୁଁ बेसिक पूरा पढ़ेइछि तमे ई बेसिक को यूज करके टिके खाली तम बही पढ़िबो ताले हेजिवो ओके
सर नमस्कार
ठीक
सर।
अछि
हाँ, नमस्कार
सर एटा मु जनवरी 2025 रे सेशन रे एडमिशन करिछि तो सर मु सेमेस्टर एग्जाम दे पारिबि तो?
तमर एई सेशन तो
आज्ञा
तमे जदि एई सेशन तले तमे देई परिबो टिके तथापि कंफर्म करनियो कोऑर्डिनेटर सर सहित कंफर्म कर
सर नंबर टा नाही तो हेति पे मु कोन माने जानि पारुनी
सर
कोनटा
1 ईयर ऊपर एग्जाम अपेयर करि परिबे आमे जेतिकी कंडीशन जो ये देखिचो।
अच्छा टिके तथापि कंफर्म हे जानतो आपनकर इग्नो साइड रे नाही कि लोकल कोऑर्डिनेटर्स के अछंती तथापि मु पचारि कि नंबर देई देबी आपन काली
आज्ञा
आपन
सर
जा
काली टिके माने
हाँ
जणे देले मु
जा
क्लियर नियंती।
ଯାହାର डाउट ଅଛି ସେ ପଚାରି ଦବ ତାଙ୍କୁ ପଚାରି ଦେଲେ ସେ କ୍ଲିୟର କରିବ
आज्ञा
थैंक यू सर।
ଆଉ ନିଲେଶ आई हैव लॉट ऑफ डाउट्स नीलेश कुमार के नीलेश कुमार के अछंती ओके
सर ये जो क्लास है मो लेट रे जॉइन हेले मो जॉइन हेलानी ताले यार किछि माने रिकॉर्डेड वीडियो किछि मिलिबो कि
सेटा आपण कांटेक्ट करिवे कोऑर्डिनेटर सर संगे मु तो रिकॉर्ड करूनी से मन रिकॉर्ड करछंती से मन पठेबे
गुड इवनिंग सर।
हां गुड
सर
इवनिंग
काइंडली आपण जो सिलेबस टा प्रेजेंट करछन्ती सेटा टिके हम सह शेयर कर देइथे हमें भी टिके पाई जाइथांतु।
मु तो ग्रुप रे नाही मते पिंग कर दियो सेतरे शेयर हे जी
ओके थैंक यू सर।
ସାର୍ ଆପଣଙ୍କର ଏଇ ନମ୍ବରରେ ହ୍ୱାଟ୍ସଆପ୍ ଅଛି ଏଇ ନମ୍ବରରେ ମାନେ ମେସେଜ କଲେ ଆପଣ ଦେଇଦେଲେ ଗ୍ରୁପରେ ମୁଁ ସେୟାର
हाँ
କରିଦେବି
हाँ
ସମସ୍ତଙ୍କ
हाँ
ସହିତ।
हाँ हाँ ମୁଁ
ହଁ ମୁଁ
ଦେଇଦେବି
ମେସେଜ କରିଦଉଛି ଆପଣଙ୍କୁ।
ଠିକ ଅଛି ମତେ ପିଙ୍ଗ କରିଦିଅ ମୁଁ ସେଇଥିରେ ପଠେଇଦେବି
ହଁ ସାର୍ ଥ୍ୟାଙ୍କ ୟୁ।
ଆଉ କାହାର ଡାଉଟ ଅଛି
सर मोर डाउट अछि
ହଁ କୁହ
सर मु फर्स्ट सेमेस्टर रो असाइनमेंट्स जाको सबमिट करि सारली सर
ହୁଁ
आउ सेकंड सेमेस्टर रो एबे आउथरे सबमिट करि परिबे सर एबे जस्ट आज ही सबमिट करिछि मु तो सेकंड सेमेस्टर रो सबमिट करि परिबे सर आउथरे
एक्चुअली ये जो असाइनमेंट ଆଉ ଏ ଯୋଉ रिलेटेड जदि किसी डाउट अछि मु काली पचारि के आपनको नंबर ଦେଇଦେବି ସାର୍ जी अछंती तांको आपण कांटेक्ट करदेबे सेठी आपनकर जहा जहा भी डाउट थिबो एवडा मो नॉलेज रे नाही मते खाली क्लास में असाइन कर
ये सर अगेन
सर
सबमिट
हमर
करे हबो लास्ट डेट जाए
कि आपनकर जो ग्रुप अछि ग्रुप रे नांती कि सेठी अन्य फ्रेंड माने आपनकर भी जानिथिबे ना
सर
बट
ऑलरेडी सर मु काली ही सबमिट करछि गोटे सेमेस्टर तो आउ थरे लास्ट डेट जाए सबमिट करी हबो सेकंड सेमेस्टर असाइनमेंट गुडा मु सारन से तो କଥା ही के आसीछि
जे କଥା ହେଇचंती से सारंग सहित डिटेल्स କଥା ହେଇथिबे तांको कांटेक्ट करो
आज्ञा
तम
सर
भितरे ग्रुप भितरे कांटेक्ट करो
ସାର୍ ଆମର କେନ୍ଦ୍ରାପଡ଼ା ଷ୍ଟଡି ସେଣ୍ଟରରା ମାନେ ସଣ୍ଡେ ୱିଦାଉଟ୍ ସଣ୍ଡେରେ ଅନ୍ୟ ଦିନରେ କେତେଟାରୁ କେତେଟା ଟାଇମ୍ ଭିତରେ ଖୋଲା ହଉଛି ମାନେ
ମୋ ନଲେଜ ରେ ଏଗୁଡାକ କିଛି ବି ନାହିଁ
ଆଜ୍ଞା ସାର୍
ମୁଁ କହିଲି ନା ଆପଣଙ୍କୁ ନମ୍ବର ଦେଇଦେବି ଆପଣ ଖାଲି କଥା ହେଇଯିବେ डिटेल्स
ଆଜ୍ଞା
ଆଠଟାରୁ ଦିଟା ଖୋଲା ରହୁଛି।
ଏଇ
ମାନେ
ଦେଖ
ଅନ୍ୟ ଦିନ
ତମ
ଗୁଡାକରେ ନା ରବିବାରେ କେବଳ?
କେବଳ ରବିବାର 8ଟାରୁ 2ଟା ଖୋଲା ରହୁଛି, ଅନ୍ୟ ଦିନ ସକାଳ 8ଟାରୁ 10ଟା ଯାଏଁ ଖୋଲା ରହୁଛି। ଆଉ ଅନ୍ୟ ଦିନ ତୁମେ ମାନେ ଫିଜିକାଲି ସବମିଟ କରିପାରିବ ନାହିଁ, କେବଳ ସଣ୍ଡେ ହିଁ କରିପାରିବ।
ମଣ୍ଡେ ରୁ ଫ୍ରାଇଡେ 7ଟାରୁ 10ଟା ଖୋଲା ରହୁଛି, ଆଉ ସଣ୍ଡେ ଦିନ 8ଟାରୁ 2ଟା।
ଫିଜିକାଲି ଅନ୍ୟ ଦିନେ ସବମିଟ କରିହବ ନାହିଁ ନାଇଁ?
ଅନ୍ୟ ଦିନେ ନଉନାହାନ୍ତି ସେମାନେ ଫିଜିକାଲି।
ଠିକ ଅଛି, ଥ୍ୟାଙ୍କ ୟୁ ଥ୍ୟାଙ୍କ ୟୁ।
ଆପଣ ଚାହିଁଲେ ସ୍ପିଡ ପୋଷ୍ଟରେ ବି
ଓକେ
ପଠେଇ ପାରିବେ, ମୁଁ ସ୍ପିଡ ପୋଷ୍ଟରେ ପଠେଇଛି। ମାନେ ସକାଳ 8ଟାରୁ ସକାଳ 10ଟା କି?
ଗୁଡ୍ ଇଭିନିଂ ସାର୍।
ଗୁଡ୍ ଇଭିନିଂ।
ସାର୍ ଆକ୍ଚୁଆଲି ମୁଁ ଟିକେ ଲେଟ୍ରେ ଜଏନ୍ କଲି ସାର୍। ଆଜି ଯୋଉ ଆପଣ ଯୋଉ କ୍ଲାସଟା ଯୋଉଟା କରିଛନ୍ତି ସେଟାର କିଛି ରେକର୍ଡିଂ ମାନେ ଆଭେଲେବୁଲ ହଉଛି କି?
ରେକର୍ଡିଂ କିଛି ନାହିଁ, ମୁଁ ରେକର୍ଡିଂ ଆପଣ କଣ୍ଟାକ୍ଟ କରିବେ ଆପଣଙ୍କର ଯିଏ ଅଛନ୍ତି କୋଅର୍ଡିନେଟର ସାର୍ ଅଛନ୍ତି ତାଙ୍କ ସହିତ କଣ୍ଟାକ୍ଟ କରିବେ, ଅର୍ ଆପଣ ମୁଁ ପଠେଇଦେବି ପିପିଟି
ଓକେ ସାର୍।
ସେୟାର ବି ଯୋଉ ଗ୍ରୁପରେ ହେଇଯିବ, ଆପଣ ପିପିଟି କି ଥରେ ରିଡ୍ କରନ୍ତୁ, ଆପଣ କ୍ଲିୟର ହେଇଯିବ।
ଓକେ ସାର୍, ଓକେ ସାର୍, ଥ୍ୟାଙ୍କ ୟୁ ସୋ ମଚ୍ ସାର୍, ଓକେ ସାର୍।
ହୁଁ।
ସାର୍ ୟା ପାଇଁ ଯୋଉ ଇଗ୍ନୋର ପଢା ହଉଛି, ୟାର କିଛି ମତଲବ ୟୁଟ୍ୟୁବ ରେ କିଛି ସୋର୍ସ ଅଛି କି ଯୋଉଥିରେ ଆମେ ଯଦି ବୁଝି ନ ପାରିଲେ ଇଏ କଲେ କିଛି ସୋର୍ସ ଅଛି କି ତା ଦେହରେ?
ହଁ, ୟୁଟ୍ୟୁବରେ ସୋର୍ସ ନାହିଁ। ମେ ବି ତମେ କଣ୍ଟାକ୍ଟ କରିବ ତମ ସାରଙ୍କ ସହିତ, ଯଦି ସେ ରେକର୍ଡିଂ ଦେବେ ତାହେଲେ ତମେ ସେଟାକୁ ବି ପଢିପାରିବ।
ଆଜ୍ଞା ଆଜ୍ଞା, ଧନ୍ୟବାଦ ସାର୍।
ଆଚ୍ଛା, ଭୁବନେଶ୍ୱର ଷ୍ଟଡି ସେଣ୍ଟରରେ କିଏ ଅଛନ୍ତି କି ଏଠି?
ମ୍ୟାଡାମ ଭୁବନେଶ୍ୱରରେ ତ ଷ୍ଟଡି ସେଣ୍ଟର ନାହିଁ, ରିଜନାଲ ସେଣ୍ଟରଟା।
ହଁ, ମୋର ଆଡମିସନ ହେଇଛି ଯେ ମୋର ଷ୍ଟଡି ସେଣ୍ଟର ଟା ସେଇଠି ଭୁବନେଶ୍ୱର ଦେଖଉଛି ତ ମୁଁ ଟିକେ ମାନେ
ଆପଣ
କନଫ୍ୟୁଜ।
ଏଇ ଜୁଲାଇ 2020 ଏଇ ଯୋଉ ଜାନୁଆରୀ 2025 ରେ କଲେ?
25ରେ କରିଛି ମୁଁ ଏବେ ଆଡମିସନ।
ମେ ବି ଆପଣଙ୍କର ଚେଞ୍ଜ ହେଇଯିବ, ଆମ ମାନଙ୍କର ତ ସେଇ ଭୁବନେଶ୍ୱର ଦେଇଥିଲୁ କିନ୍ତୁ କେନ୍ଦ୍ରାପଡାକୁ ଚେଞ୍ଜ ହେଇଯାଇଛି।
ମୋର ସେଇଟା ଦେଖଉନି ତ,
ସାର୍ ଆଜି
ମାନେ
ପାଇଁ
21
କ୍ଲାସ୍ ଏତିକି।
ଦେଖଉଛି ତ ମୁଁ କନଫ୍ୟୁଜ ପୁରା।
ହଁ, ଆଜିର କ୍ଲାସ ଏତିକି। ନେକ୍ସଟ ଆମେ କାଲି ଷ୍ଟାର୍ଟ କରିବା ଆମର, ମୁଁ ଯୋଉଟା ଆପଣଙ୍କୁ କହିଲି ଟପିକ ଟା ସେଲ୍ ଟ୍ରାନ୍ସପୋର୍ଟ କାଲି ଡିସକସ କରିବା, ଆଉ ମୁଁ ଆଜି ବି ପିପିଟି ଆପଣଙ୍କୁ ସେୟାର କରିଦେବି ଯାହା ପଢେଇଲି, ଯଦି ରିଡ୍ କରିପାରିବେ ତ ବେଟର ହବ।
ଆଜ୍ଞା ସାର୍, ଥ୍ୟାଙ୍କ ୟୁ ସୋ ମଚ୍।
ଆଜ୍ଞା ସାର୍, ସାର୍
ସିଲାବସ
ନମସ୍କାର
ଟା
ସାର୍,
ଏ ପିପିଟି ଟା
ଥ୍ୟାଙ୍କ ୟୁ
ଦିଟା
ସାର୍।
ଉଭୟଟା
ମୁଁ
ଦରକାର।
ସାର୍ କୋଶ୍ଚିନ୍
କଣ କହିଲେ?
ଆଜ୍ଞା
ସିଲାବସ
ସାର୍
ଟା
ଲାଷ୍ଟ
ପିପିଟି
ସାର୍
ଟା
ଗୋଟେ
ଦିଟାଙ୍କ।
ହଁ ହଁ, ମୁଁ ସେୟାର କରିଦେବି। ହଁ, ଆଉ କିଏ ଜଣେ
ଆଜ୍ଞା
ପଚାରୁଥିଲେ?
ଆଜ୍ଞା ସାର୍, ନମସ୍କାର ସାର୍। ସାର୍ ଆକ୍ଚୁଆଲି
ହୁଁ
ଫାଷ୍ଟ ଯୋଉ ଆମର ସାଇଟୋସ୍କେଲେଟନ ଫାଷ୍ଟ ଯୋଉ ଚାପ୍ଟର ଟା ପଢେଇଲେ ସେଟା ମୋର ଟିକେ ଡାଉଟ କ୍ଲିୟର ହେଇଯାଇଛି, କିନ୍ତୁ ସାର୍ ଯୋଉ ଆମର
ହୁଁ
ରହିଲା ଆକ୍ଟିନ ଫିଲାମେଣ୍ଟ, ତାର ଯୋଉ ପ୍ରୋଟିନ ବାଇଣ୍ଡିଂ ଟା ବହିରୁ ଥରେ ପଢିଥିଲି ମୁଁ ଟିକେ ଜାଣି ପାରିଲିନି। ଖାଲି
ହୁଁ
ଯେହେତୁ ଆଜି ଫାଷ୍ଟ କ୍ଲାସ ଥିଲା, ଟିକେ ମୁଁ ମାନେ ଡାଉଟ ଟା ପଚାରି ପାରିଲିନି। ଟିକେ ସାର୍ ପିପିଟି ଟା ସେୟାର କରିଦିଅନ୍ତୁ, କାଲି ଟିକେ ଫାଷ୍ଟ ଟିକେ ଆମକୁ ଟାଇମ ଦେବେ ଡାଉଟ ଗୁଡା କ୍ଲିୟର କରିଦବା ରିକଲ କରିକି, ତାପରେ ଆମେ ନ୍ୟୁ ଚାପ୍ଟର ଷ୍ଟାର୍ଟ କରିବା ସାର୍। ସାର୍ କଣ
ଠିକ ଅଛି
ପ୍ରୋବ୍ଲେମ?
ଠିକ ଅଛି।
ଓକେ ସାର୍,
ନା
ଥ୍ୟାଙ୍କ
ନା
ୟୁ।
ହେଲା, ଓକେ।
ପ୍ରାକ୍ଟିକାଲ ଏକ୍ସାମ ର ଡେଟ୍ କିଛି କହିନାହାନ୍ତି?
ନା ନା, ସବୁ କୋଅର୍ଡିନେଟର ଆପଣଙ୍କ ରିଜନାଲ ସେଣ୍ଟର ହେଡଙ୍କୁ ପଚାରିବେ, ସେଇଠି ଆପଣଙ୍କର ସବୁ ସଲ୍ଭ ହେଇଯିବ।
ସାର୍ କାଲି ଟିକେ ନମ୍ବର ଟା ସେୟାର କରିଦେବେ କୋଅର୍ଡିନେଟର ସାରଙ୍କର।
ହଁ, ମୁଁ ପଚାରିକି ସେୟାର କରିଦେବି।
ଆଜ୍ଞା ଆଜ୍ଞା, ଥ୍ୟାଙ୍କ ୟୁ। ଥ୍ୟାଙ୍କ ୟୁ ସାର୍।
Okay,
ଥ୍ୟାଙ୍କ ୟୁ ସାର୍।
we can end the session.
ଥ୍ୟାଙ୍କ ୟୁ ସାର୍।
Okay, thank
ଥ୍ୟାଙ୍କ ୟୁ ସୋ ମଚ୍ ସାର୍।
you. Thank you.
ସମସ୍ତେ ସେସନ ମ୍ୟୁଟ୍ କରିଦେଲେ କି? हेलो। हेलो समझ क्वेश्चन लीव कर देले कि
जनवरी 2025 बैच रे की अछंती रिप्लाई करंतु 2025 जनवरी बैच रे की अछंती रिप्लाई करंतु
इटा मैडम मु जॉइन करिछि जनवरी 2025 रे।
अच्छा तो तार किसी सेपरेट ग्रुप अछि की
नहीं सेटा तो मु भी खोजुछि कोडी ग्रुप अछि की बेली।
ठीक अछि तो आपन के ई यार लिंक कोडू मिलला सेई जुलाई 2025 2024 बैच
हाँ
रू
हाँ हाँ हाँ
सेथरे को मेसेज करथि के रिप्लाई देले ठीक अछि।
ସାଉଣ୍ଡ କିଛି ଆସୁନି ସାର୍। କିଛି ଶୁଭାଯାଉନି ସାର୍।
7:36 6ଟା ଖାଇଦେଲି ପେଟ କଣ ହେଇଗଲା। ଦେ ପଠେଇଲି ଦେ ଭାତରେ ଦେଲୁ ପଖାଳି ଦିଏ କାଇଁ ରହିବ ନାଇଁ ପୁଣି ଏତେ ଲେଖି ସାରିଲା ପରେ ପୁଣି ପିଡିଏଫ୍ ବନା ହବ। ସାତଟା 37 ହେଲା। ଦେଖିଲୁ ଏତେ ସବୁ ଲେଖିଦବୁ ତୁ ଏଇଟା ତୋ ଦାୟିତ୍ୱ ସବୁ ଯାକ ତୁ ମତେ କାଲି ସୁଦ୍ଧା ଲେଖିଦବୁ କାଲି ରାତି 10ଟା ସୁଦ୍ଧା 39 ପେଜ। ନାଇଁ ଡାଉନଲୋଡ କରିଛି। ଲେଖି ସାରିଲା ପରେ ପହରଦିନ ସକାଳୁ ସବୁ ପିଡିଏଫ୍ କରିବି। ଫୋନ ତୁ ଯିବୁନି ନା ତୁ ଗୋଟେ କାମ କରିବୁ ଖାଲି ଫଟୋ ଉଠେଇବୁ ପିଡିଏଫ୍ ମେକରରେ ଚିତ୍ର ବି ହବ। ସେମିତି ଖାଲି ଲେଖା ହବ ବ୍ଲୁ ପେନ୍ ଆନ୍ସର କିଛି ନାହିଁ। ପାଗଳ ହେଇଗଲି ସେ ଡାଟା କଲେକ୍ଟ କରିବାକୁ ଲେଖିବାକୁ। ବାପା କହିଦେଲେ ଆମର ଉମର ମନ୍ଦିର ଉପରେ ନା ଭଲ ହବ ଲେଖିବା ପାଇଁ ତେଣୁ ମୁଁ କହିଲି ଉମର କହୁଥିଲି ଅଲଗା କୋଉ ମନ୍ଦିର ଉପରେ ନବା ପାଇଁ କାହିଁକିନା ସେ ବିଷୟ ଦଉନାହାନ୍ତି ସେ କହୁଛନ୍ତି ତମେ ଗୋଟେ ଜାଗାକୁ ଯିବ ମୋ ସାଙ୍ଗରେ ପେପର ନେଇ ଆସିବୁ ଏଇଠି ଲେଖିବୁ ନା ଘରେ ଲେଖିବୁ ଆସୁନୁ ଘରେ ଲେଖିବୁ ବର୍ଷାକୁ ଦେଇଛ ଲେଖିବୁ ମାନେ ନେଇଥିଲା କହିଲା ମୁଁ ଆସିବି 8:30 ବେଳେ ଫୋନ କରିଥିଲ ମତେ କହିଲା 8:30 ମୁଁ ଆସିବି 2 ଘଣ୍ଟା ଲେଖିବି ତାପରେ ଏଇଠି ଶୋଇବାକୁ ଆସିବି ସେ ଆଜି କାଲି ଯଦି ତମେ କହିଥା ଆସିଥାନ୍ତ କାଲି